All right, can everyone see my slides and hear me well? Okay, great. I really appreciate this opportunity to share my, the postdoc work. Actually, most of the work definitely was accomplished at MIT. So basically, during my time at, at MIT, we were working on this, developing a really synthetic vaccine to further improve CAR T cell therapy. And after the first piece of work, and then we realized actually the vaccine boosted CAR T therapy can crosstalk with host immunity to really trigger a very potent anti-tumor endogenous immune response to deal with the residual tumor cell. And this eventually we discovered that actually can be harnessed to directly treat tumors with pre-existing antigen heterogeneity, which seems to be true for most of solid tumors. Okay, so before talking about vaccine or CAR T, I want to briefly give a little bit overview of cancer immunotherapy based on T cells. I know Olga already gave a very good introduction here, but here I just want to kind of a little bit maybe repeat a little bit. So just make sure we're on the same page. So basically, kind of within the solid tumor, you can discover many of the different kind of immune cell types, such as macrophage, NK cells, and DCs, different type of T cells and neutrophils. And among this kind of immune cell population, the macrophage or neutrophil may be kind of towards a bit immunosuppressive. NK cell, T cells can be directly killing the tumor cells, and the dendritic cell will be amplifying the immune responses. And a successful immunotherapy is typically achieved by balancing the number and functionality of these different type of immune cells. And talking about these different immune cells, one of the major immune cell type here is CD8 T cell, which is responsible for directly recognizing and killing tumor cells by secreting a variety of these effector molecules such as granzyme, perforin, and interferon gamma. And so based on a variety of studies, if you directly deplete CD8 T cells from a very successful immunotherapy, and then you almost completely lose the immunotherapeutic efficacy. So this really highlights the super important role of CD8 T cells in tumor control. So T cells recognize tumor cells through a very unique mechanism. So T cells have their own kind of T cell receptor, which is kind of actually multimeric complex. And then this TCR recognizes the target, which is a small peptide that's only presented in complex with another protein called MHC on tumor cells. And this interaction is very, very sensitive. It can recognize even just one single peptide molecule there. And this very high sensitive interaction eventually triggers some interest in the scientific community to harness this phenomenon. Potentially, you can turn a non-tumor reactive T cells into a tumor reactive population by just genetically engineer them by transducing them with these tumor specific TCRs. And the advantage of this, of course, you have this ultra high sensitivity, and you have this flexibility. Essentially, can program a T cell target any desired tumor cell type bearing this tumor antigen. However, there's a practical barrier to translating this type of therapy, which is MHC restriction and MHC polymorphism. Because every single individual here, we roughly have 6 different MHC one allele and 6 different and MHC 2 alleles. And basically, for patients bearing these different specific antigens, if you want to match the specific antigen and MHC allele that can present this antigen, now the number of patients can really benefit from this therapy is significantly reduced. So here it actually comes why the CAR T cell have been really promising, because it really bypasses this MHC dependent targeting and make it MHC independent. And the CAR T cell is engineered in a way that to synthetically kind of connecting a antigen recognition domain from antibody to the signaling domain from a T cell receptor complex. And now you have a synthetic molecule essentially making an orthogonal sensor on the T cell, allowing the T cell to recognize something a typical antibody can recognize. So this essentially broadened the target recognition by the T cells and allows it to recognize tumor cells without worrying about this MHC polymorphism. So this is just showing some clinical outcome from the early stage clinical trials. So on the left, you can see this is the CD19 CAR T, which have already been approved by FDA. And when the patient bearing this bone marrow a blast, and once they receive this CAR T treatment, within a month, you can see almost all their bone marrow blasts got cleared, which means they have gotten rid of all their leukemic cells. And more than 70% patient can really benefit this to achieve long-term survival. And right now, FDA have already approved 6 different CAR T products, 4 targeting leukemia lymphoma, the other 2 targeting multiple myeloma. So this is for the anti-leukemic CAR T. But for the anti-solid tumor CAR T, this is just one example I'm showing on the right. This is actually one of the clinical trials carried out here at the University of Pennsylvania. So this is one type of solid tumor called glioblastoma. This glioblastoma is a brain tumor. Many of them expressing EGFR3 oncogene on the tumor cell surface, just making them a highly unique surface marker for this type of tumor. But because this antigen is not homogeneously expressed all solid tumors and also have significant inter-patient heterogeneity, therefore you can see like this type of antigen-specific CAR T therapy only leaves like probably 10% patients having this long-term survival. And the reason they have this like very low like long-term survival just because most other patients relapse with antigen-negative tumors. So now kind of we're talking about like how to deal with these hurdles. So in order to to deal with the hurdles of CAR T therapy in solid tumors, we first want to understand the difference between the successful CAR T therapy in leukemia lymphoma setting versus solid tumor. So as you can see here in this depiction, in the leukemia lymphoma setting, the CAR T cell essentially coexist with their target cell. So that means once they got infused into patient, they will be in co-circulation with the target cell. Therefore, they can be constantly interacting with the target cell, receiving antigen stimulation, and achieving this like early stage expansion. And this early stage expansion is super important for them to starting like kind of engrafting and developing these memories. And also, they can benefit from this regenerating healthy B cell from bone marrow as well. However, for solid tumor, as you can see, most of the antigen really kind of localized in this like little confined area, which is also highly immunosuppressive. And this limited antigen exposure and immunosuppressive tumor marker environment really leads to limited CAR T expansion, persistent functionality, and eventually compromise their overall therapeutic efficacy in solid tumors. And there are, of course, additional hurdles, such as lack of tumor-specific surface antigen, and also the antigen loss or antigen tumor heterogeneity. And this is actually the histology that I mentioned about this EGFR3 expression in glioblastoma. You can really see they have significantly variable kind of antigen expression across different patients. So that's why kind of one of the major issue for CAR T therapy in solid tumors. So initially, when we think about the question, we just like very interested in developing a vaccine just to address this CAR T expansion persistence issue. But then, kind of later on, when the project progresses, we realize this can actually can partner with endogenous immunity to deal with this tumor heterogeneity issue. So now let's come to this vaccine. So before talking about the synthetic vaccine, I want to briefly touch upon how the conventional vaccine works. So in the conventional vaccination setting, you typically will formulate your antigen with your adjuvant and then inject into the patient. And then this protein antigen and adjuvant will co-drain through lymphatics into the lymph node, and where this antigen will be captured by this lymph node resident antigen presenting cell, which can process the protein antigen into small peptide, and then eventually the small peptide can be loaded onto these empty MHCs, eventually presented to T cell through this peptide MHC TCR interaction. And this essentially defines the T cell response specificity and serves as signal one. So, but the adjuvant also will drain to lymph node and then stimulate antigen presenting cells to let them become activated. Once the APC got activated, they will operate with signal 2 and signal 3, which is co-stimulation and cytokine support. So only in the presence of all three signals, you will see complete T cell activation and optimal T cell differentiation and proliferation. So now how can we borrow this concept to develop such a vaccine for CAR T cells? And the first thing we have to understand is how the CAR T cell actually operates, because the CAR T recognized target is in a different way compared to TCR. So the CAR recognizes the antigen that's directly displayed on the cell surface instead of like presented in complex with MHC. So therefore, this requires us to directly display the ligand recognized by CAR on the APC, then which can efficiently trigger CAR T. To achieve this goal, we tend to a amphiphyal polymer, so we call it amphiphyal ligand molecule. So this amphiphyal ligand molecule have three components. The first component is called the lipid tail, which have two functions. One is binding to cerebral albumin, the other is directly inserting into the lipid bilayer of any cell. And then the second component is PEG linker, which essentially helps solubilization of this molecule. And third component is essentially any of the ligand that you are interested for the CAR to recognize. And typically, when you are mixing this amphiphyal polymer with, I would say, in an aqueous solution such as PBS, they will form a small nanoparticle called a micelle. But once you inject it in vivo, they will encounter cerebral albumin, which have strong affinity toward this lipid tail. Then they can break apart into, basically this particle can break apart into monomer and be carried out into the adrenal lymph node. And once they enter the renal lymph node, this lipid polymer can dissociate from this albumin and then jump into the surface of APC and eventually present it to the CAR T in a synthetic way, together with co-stimulation and pro-inflammatory cytokine. So this is the concept. So let's see how it actually works. So in order to model this, we initially focused on a fluorescent labeled amplifier molecule called amplifier 50, and then we did like a tail-based injection. Basically, you can track where this molecule traffics to. And you can see most of the signal localized actually within the immediate renal lymph node called inguinal lymph node, and then slightly distal external lymph node. And we also kind of isolate the cells from this lymph node, and you can see most of the cells that capture this molecule have this molecule display on their cell surface, just like a ring structure. And we also analyze like other organs that the molecule can potentially traffic to. But what we discovered that almost 99% of signal is really kind of localized in the adrenal lymph node. So this really shows the specific lymph node targeting and cell surface incorporation property, which is exactly what we expected. So now once we know like the molecule traffic the lymph node, we also want to dive in deeper to understand which region of the lymph node actually this molecule traffics to. So based on the confocal imaging, you can see most of the 50 signal, which is essentially amplifier 50 molecule, are localized within this interfollicular region, which is exactly the region many of the antigen presenting cells are localized. But we also showed that using this dissociated individual cell population from the lymph node to analyze which specific population actually captures this. And as you can see here from this effect plot, like most of the population that capture this molecule and display the 50 molecule on the surface are these conventional APCs, such as microphage, medullary macrophage, or C11C, C11D double positive DCs. However, for other cell population with the lymph node, they only have minimal capture of this polymer and presentation. Okay, so now we have been talking about like this model amplifier molecule, and this now we want to see if we can develop a bona fide vaccine for CAR T cells that recognizing tumor cells. So we focused on this EGFR-3 glioblastoma, and for multiple reasons, because this EGFR-3 is a highly specific tumor surface antigen that we can develop a CAR T for, and there are available monoclonal antibody sequence we can generate a CAR against. And so what we did here, we took this minimal epitope that recognized by the CAR and put it onto our lipid polymer, and then using this antibody information to produce this CAR T cells. And then we co-culture a cell labeled with this amplifier peptide 3 ligand, and then stimulate the CAR T, and we can see very strong and potent stimulation in vitro. So this shows, at least in vitro, this platform works pretty well. And then we move on in vivo. What we did here is we established tumor in the mouse model, and then give them lymphodepletion, which is typical treatment regimen before you giving the CAR T in the patient, and then followed by CAR T injection, and then followed by single dose of vaccination. So what you can see here is that even though the mice bear tumor, but with just CAR T, you do not see much of CAR T expansion in the periphery. But with single dose of vaccination, you can see very potent CAR T stimulation, CAR T expansion, and which can last for more than 3 weeks. And we also sample the peripheral blood for analysis, basically characterizing their functionality and phenotypes. And what you can see here is that with vaccine boosting, these CAR T cells have increased cytokine secretion, increased mitochondrial mass, and increased granzyme B, which essentially represent increased polyfunctionality of the CAR T of these CAR T cells. And throughout all this data, it essentially shows that with vaccine boosting, we're not just getting more CAR T cells, but also getting better CAR T cells. And then we also want to, so the analysis we did just now was all in the periphery blood, essentially. Here we want to see what actually happens within the tumor. We basically also set up tumor-bearing mice and injecting CAR T cells followed by vaccine boosting. And then we took out the tumor, analyzing how many CAR T cells actually get in the tumor and what's their phenotype. So based on this data, you can see with the vaccine boosting, you can see almost like 10-fold increase of CAR T infiltration into solid tumor. And this infiltrated CAR T cells also exhibited increased like proliferation, which is shown by this increased KLC7 staining, and also increased polyfunctionality, such as granzyme B, interferon gamma, and TNF alpha. And we also perform like RNA sequencing to profile the CAR T cells from the vaccine boosted or non-boosted mice. And interestingly, there's very kind of strong signature associated with proliferation, such as MYC or E2F, which essentially correlates with our KLC7 staining here. But another major signature here is actually metabolic reprogramming. You can see almost like all 4 signatures associated with this are associated with T cell metabolism, such as this oxidase phosphorylation, mTOR C1 signaling, fatty acid metabolism, and peroxisome signaling. And actually, among all of this, oxidative phosphorylation had already been shown to improve CAR-T persistence and overall anti-tumor functionality. So this also means that this vaccine boosting also kind of helps sustain the CAR-T metabolism within the solid tumor to hopefully to maintain their enhanced anti-tumor efficacy. And of course, when we're monitoring the overall tumor growth, you can see with the vaccine boosting, the overall tumor growth is much more slower, and also roughly 60% of animals can be cured with CAR-T and a vaccine boosting. So till this point, I would say we have very solid data showing this vaccine boosting can really enhance CAR-T therapy, and this. So we thought this kind of the end of the story, but actually when we are really diving into the endogenous immunity, which actually one of the reviewer asked that question when we're doing the first paper revision, and then we discovered this. So this actually associated with highly associated with the endogenous immunity is called antigen spreading. So that means when tumor being killed by CAR T cells, there will be tumor debris being generated, and then this tumor debris can be captured by migratory dendritic cells, which can carry the debris into the adrenal lymph node to continuously priming the endogenous anti-tumor T cells. And once these endogenous anti-tumor T cells being activated, and they will come out of the lymph node and enter the tumor again to deal with the residual tumor cells. So in order to really monitor this process, we perform infrared gamma ELISpot using the endogenous T cells we isolated from the mouse. And so what you can see here is that we really try to set up like different combination of our vaccine. So vaccine, so basically here, if we just label vaccine, that means a complete vaccine, which is essentially a combination of this antiviral ligand and an effective adjuvant. Or if we kind of separate these two different components, and what you can see here is really have not much effect on the CAR T cell therapy-induced antigen spreading. But only in with the complete vaccine, you can really trigger very potent antigen spreading. And also with CAR T cell itself does not have much effect, which is also seemed to be true based on the previous report. So this really shows with the complete vaccine, and we can really engage CAR T cell with the endogenous immunity. But even though we can engage this, but what's the point here? Does this really make a difference? So here we basically, to start checking this, to asking this question, whether this endogenous T cell can have a major impact in the long-term tumor control. So the way we did this is we performed CAR T plus vaccine therapy in two different mouse models. One is wild-type mice, which means they have the normal antigen spreading because they have the healthy T cell there. The other model is regular knockout mice, which do not have B cell or T cells, therefore they do not have antigen spreading. So what you can see here is that even though at some point in both mouse models, like they have the tumor reach a undetectable level, but in the wild-type mice, as long as the tumor initially being fully controlled, they never rebound. But in the regular knockout mice, which they do not have antigen spreading, and majority of the mice, even though initially they have very strong tumor control, now most of them start kind of relapse. And many of the relapsed tumor actually started showing reduced this EGFR variant expression. That means they are less likely being recognized by the CAR T anymore. And without endogenous T cells that can target tumor through a different mechanism, now this tumor will completely lose control. And now we know that this antigen spreading and the endogenous T cell response is highly important for control tumor in the long term by dealing with those antigen loss tumor. What we want to achieve kind of in this particular experiment is basically to see if we can harness the phenomenon to directly treat tumors with significant level of pre-existing heterogeneity. So the way we model this is we mix tumors that bears the antigen targeted by the CAR with the tumors does not express the antigen at different ratio, and then implant the tumor into either wild-type mice or regular knockout mice. So based on this data, you can see is that in regular knockout mice, no matter how few of the antigen-negative tumors we implant, the tumor will relapse. That means you really need this like endogenous immunity to achieve long-term tumor control. However, in the wild-type mice, even up to 20% of the tumor are antigen negative that cannot be recognized by the CAR T, but the antigen spreading seems to be enough to control the tumor growth, at least like for 50% of them. And even for when the tumor bearing higher percentage of the antigen negative tumor, the CAR T plus vaccine therapy still have some effect. So this experiment really shows like this vaccine boosting and the ability for us to engage both CAR T and endogenous immunity is super, super important to control heterogeneous tumors. And then, the next point we're trying to figure out is what's the mechanism here? we have done quite a few different studies before reaching this point, but this is essentially the killer experiment showing that if you block different kind of cytokines, especially interferon gamma here, you can really kind of completely abolish antigen spreading. So initially we thought this, we are using ELISpot and potentially using this anti-interferon gamma antibody to coat the plate for it to perform the assay. That could be just like technical, like false positive. But actually, eventually we kind of start using this interferon gamma deficient CAR T cells, which we don't have to use blocking antibody anymore. And now this kind of fully validates our observation. This really shows CAR T derived interferon gamma plays a central role here in triggering antigen spreading. And then we performed a series of studies, which hopefully, I don't think I have enough time to describe every single one of them. But what I want to do here is quickly go through to show you what the mechanism we eventually kind of discovered. And this actually, we discovered a positive feedback loop here. The CAR T will be the major producer of interferon gamma. And then interferon gamma can act on the intratumor DCs. And DC, once it got activated by interferon gamma, they can secrete another cytokine called IL-12. And this IL-12 can act back on CAR T. And this positive feedback essentially seems to is essential to maintain the CAR T metabolism. And we also showed that this major transcription factor called PGC1 alpha, which is essential for maintaining this oxidative phosphorylation in CAR T cells, also got significantly upregulated. And once this oxidative phosphorylation is upregulated, now they can produce more interferon gamma, which can continue this kind of fueling basically fueling this positive loop here and to propagating this antigen spread effect. Okay, now this is going to be a quick summary showing that the first study we have is we developed this amplified polymer vaccine, and we showed that the vaccine can be carried by albumin through a hitchhiking mechanism into the adrenal lymph node, where they can jump on the surface of APC and then trigger CAR T activation in vivo, just like how the natural T cell being activated. And this activated CAR T cells can have increased functionality, can kill tumor and control tumor more effectively. And finally, we show in the second study, this interesting phenomenon called antigen spreading. Basically means like the CAR T can kill tumor, generate tumor debris, and the tumor debris will actually be carried by a dendritic cell into the adrenal lymph node, which can further prime endogenous T cells to attack tumor through the traditional TCR peptide MHC mechanism. And this entire process is actually maintained through this CAR T derived interferon gamma and the DC derived IL-12 through this positive feedback loop here. And so, okay, so this is my acknowledgement, and this is my current team and my funding source, and we are also looking for postdoc and motivated PhD candidates.
Amazing! You just finished right on time.
Yeah.
Thank you very much, Leyuan. Thank you very much. So, let's start with a round of questions. So. First question from Rogelio, and so he said, beautiful talk.